So the blood. So blood is a special connective tissue consisting of a fluid matrix that is plasma and form elements. So plasma is a straw colored viscous fluid constituting nearly 55% of the blood. Now 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. So 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. Fibrinogens, globulins and albumins are the major proteins. Now fibrinogens are needed for clotting or coagulation of blood. Now globulins primarily involved in defense mechanisms of the body, and the albumins help in osmotic balance. Now plasma also contains small amounts of minerals like Na plus, calcium plus, magnesium plus, bicarbonate and chlorine. Glucose, amino acids and lipids are also present in the plasma, as they are always in transit in the body. Now factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factors is called the serum. So plasma without the clotting factors, that is the fibrinogens. So plasma without the clotting factors is called the serum, right? What is serum? Is plasma without the clotting factor is called the serum. Now form elements. So erythrocytes, that is RBCs, leukocytes, that is WBCs, platelets, that is thrombocytes, are collectively called form elements, and they constitute nearly 45% of the blood. Now erythrocytes or red blood cells are the most abundant of all the cells in the blood. Okay, a healthy adult man has on an average 5 millions to 5.5 millions of RBCs millimeter per cube, okay, of blood. Now RBCs are formed in the red bone marrow in the adults. Red bone marrow, these are the type of bones, okay. Now RBCs are divided of nucleus in most of the mammals and are biconcave in shape. So RBC are divided of nucleus in most of the mammals and are biconcave in shape, okay. So they have red colored iron containing complex protein called hemoglobin. Now hence the color and the name of these cells. Why it is named, why it is hemoglobin? Because heme means iron, it contains the iron, and iron probably the red in color, so this is why the color and the name of these cells like that. So healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. Now these molecules play a significant role in transport of respiratory gases, okay. RBCs have an average lifespan of 120 days, after which they destroyed in the spleen, and that is grey weight of RBC. So grey weight of RBC are the spleens, okay, are the spleen. So RBCs have an average lifespan of 120 days, okay. So leukocytes are also known as white blood cells, that is WBC, as they are colorless due to the lack of hemoglobin. So they are nucleated and are relatively lesser in number, which averages 6,000 to 8,000 millimeter per cube of blood. Now leukocytes are generally short-lived. We have two main categories of WBCs: granulocytes and agranulocytes. Neutrophils, eosinophils and basophils are different types of granulocytes. Neutrophils, eosinophils and neutrophils, lymphocytes and monocytes are the agranulocytes, and granulocytes, neutrophils, eosinophils and basophils. So these are the different types of granulocytes, while lymphocytes and monocytes, these are the agranulocytes. Now neutrophils and monocytes, these are phagocytic in nature, okay. Now neutrophils are the most abundant cells, that is 60 to 65% of the total WBCs, okay, of total WBCs, right. And basophils are the least, that is 0.5 to 1% among them. So neutrophils and monocytes, they are 6 to 8% in number, are the phagocyte cells which destroy foreign organisms entering the body. I already told you. Now basophils secrete histamine, serotonin and heparin and are involved in anti-inflammatory reactions. So when you get some kind of injury, then there is some kind of sensation, and that sensation is due to the basophils which secretes the histamine, serotonin and heparin that causes it. Okay, now the eosinophils, that is 2 to 3%, and eosinophil resist infections and are also associated with allergic reactions. So eosinophils, so they are associated with the allergic reactions, right, and they resist the infections. Now basophils, it causes kind of inflammation reactions, and eosinophil that resist it. Now eosinophils, that is 2 to 3%, right. Now lymphocytes, that is 20 to 25%, are of two major types, that is B and T lymphocytes. Now B and T lymphocytes are responsible for immune responses of the body. Now platelets also called thrombocytes, These are the cell fragments produced from the megakaryocytes. Now megakaryocytes, these are the special cells in the bone marrow. Okay. Now blood normally contains 1,50,000 to 3,50,000 platelets per meter per millimeter cube. Okay, platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. Now blood groups. So as we know, blood group of humans differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. Two such groupings, that is AB and RH, are widely used all over the world. So AB and RH, these are blood grouping. So ABO blood grouping, it has the A, B, AB and O. So ABO blood grouping is based on the presence or absence of two surface antigens, that is chemicals. Now what are the antigens? These are the chemicals that can induce immune response to produce to produce the, I mean, so that B lymphocytes can produce the antibodies against of them on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies, proteins produced in response to antigens. This was I'm talking about, like proteins produced in response to antigens. Now the distribution of antigens and antibodies in the four groups of blood A, B, ABO and O are given in the diagram. So you probably know that during blood transfusion, now when we exchange our blood, like during the blood transfusion, any blood cannot be used. Why it is so? Because the blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion, to avoid severe problems of clumping. It can happen, the clumping, that is destruction of RBCs of the receiver. Okay, the donor's compatibility is also shown in the figure. So the blood groups and donor compatibility. Okay, so the blood group is A, B, AB and O. Antigen on RBCs. Oh, A blood group has A antigen, B blood group has B antigen, AB blood group has no AB antigen, and O blood group has no antigen. Okay, so antibodies in plasma. If it is A, then antibody B is present. Now if it is anti B, so anti A is present. If it is anti A, antigen A and B, then no antibody is present. An antigen on the RBC, if it is nil, like no antigen, then antibodies in plasma is anti A and anti B. So O is the blood group. So now donors group, you can see AB is the universal acceptor. So O blood can be donated to person which any other blood group, and hence O individuals are called universal donors. So O is the donor, that is universal donor. You can, I mean, it can get to the anyone. Now persons with AB group can accept blood from persons with AB as well as the other groups of blood. Therefore such persons are called universal recipient. So A now if someone is A, so donors group can be A just like them, or O is as the universal donor, then B, so BO, AB and O can taken only by O. Okay, just remember this. Now RH grouping. So another antigen, the RH antigen, similar to one present in rhesus monkey. Hence RH is also observed on the surface of RBCs of majority, that is nearly 80% of humans. Such individuals are called RH positive. So 80% of humans we find there is an RH antigen. Now such individuals are called RH positive, and those in which this antigen is absent are called the RH negative. RH negative person, if exposed to RH positive blood, will form specific antibodies against the RH antigens. Therefore RH group should also be matched before transfusion. So there can be also the problems of the clumping. So this is why an RH negative person, because RH negative person does not have an antibodies, but if exposed to RH positive blood that has the antibodies, will no RH negative person has the antibodies. If exposed to RH positive, now RH positive does not have any antibodies, so will form. Now if it is exposed to RH positive, then RH negative person will form the antibodies against the RH antigen. Okay, which is present in the RH positive person. Okay, so therefore RH group should be also matched before transfusion. Okay, a special case of RH incompatibility, that is mismatching, has been observed between the RH negative blood of a pregnant mother with RH positive blood of the fetus. Okay, RH antigens of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the material blood to small amounts of the Rh positive blood from the fetus. In such cases, the mother starts preparing antibodies. As I said you, that Rh negative person starts making the antibodies against Rh antigen in her blood. In case of a subsequent pregnancy, the Rh antibodies from the mother that is Rh negative can leak into the blood of the fetus Rh positive and destroy the fetal RBCs. Now, this could be fatal to the fetus or could cause severe anemia and jaundice to the baby. Remember, it can cause a severe anemia and jaundice. Now, this condition is called erythroblastosis fetalis. This can be avoided by administering anti-Rh antibodies. Now, this can be treated, can be anti-Rh antibodies to the mother immediately after the delivery of the first child. Immediately after the delivery of the first child. Now, coagulation of the blood. You know, these are the platelets that causes it. So you see, like blood exhibits coagulation or clotting in response to an injury or trauma. This is mechanism to prevent excessive loss of blood from the body. Okay, so you may observe a dark reddish brown scum form at the site of a cut or an injury over a period of time. It is a clot or coagulum formed mainly of a network of threads called fibrins, in which dead and damaged form elements of blood are trapped. Called fibrins. Okay, network of fibrins and damaged form elements of blood are trapped. Okay, fibrins are formed by the conversion of inactive fibrinogens in the plasma by the enzyme thrombin, and this thrombin in turn are formed from another inactive substance present in the plasma called prothrombin. So prothrombin to thrombin by an enzyme complex is thrombokinase is required for the aberration. This complex is formed by a series of linked enzymatic reactions. These are the cascade processes involving a number of factors present in plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by the tissues at the site of injury also can initiate coagulation. Calcium ion plays a very important role in the clotting. As you can see, this is the coagulation of blood. So the limb, that is the tissue fluid. So as the blood passes through the capillaries in tissue, some water along with many small water soluble substances move out into the spaces between the cells of the tissues, leaving the larger proteins and most of the form elements in the blood. So leaving the larger protein and most of the form elements in the blood vessels. Now this fluid released out is called the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients and gases etc. between the blood and the cells always occur through this fluid. So between the blood and the cells always occur through this gland. Okay, so between the blood and the cells always occur through the fluid. So exchange of nutrients gases between the blood and the cells between the blood and the cells always occur through this fluid. Exchange of nutrients and gases, right? And an elaborate network of vessels called the lymphatic system collects this fluid. So there is an lymphatic system that collects this fluid and drains it back to the major veins, that is major veins, that opens into the right atrium, I think right atrium, right? Now back to the major veins, right? Now the fluid present in the lymphatic system is called the lymph. Because fluid is present in the lymphatic system is called the lymph. Now the lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones. Okay. Now fats are absorbed through lymph in the lacteals present in the intestinal villi. So fats are absorbed through lymph in the lacteals present in the intestinal villi. So in the lacteals present in the intestinal villi, so in the intestinal villi there is a lacteals which there is a lymph, right? In the lacteals, so lacteals present in the intestinal villi, so fats are absorbed through it. Now the circulatory pathways. Now the circulatory pathways so the circulatory pathways are of two types, that is open and closed. So open circulatory pathway is present in arthropods and mollusks, in which blood pumped by the heart passes through the large vessels into open spaces or body cavities called sinuses. So body cavities are the sinuses, okay, through large vessels into open spaces or body cavities called sinuses. Now annelids chordates have a close closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of blood can be more precisely regulated. Right now, all vertebrates possess a muscular chamber heart. All vertebrates, no matter if they are fishes and amphibians, reptiles, and birds and mammals, so all of them they possess a muscular chamber heart. Now, fishes have a 210 chamber heart with an atrium and a ventricle. So all these possess like so all they are like so all these also all these are like I mean close circulatory system, right? Now fishes have two chambered heart with an atrium and a ventricle. Now amphibians and the reptiles except crocodiles have a three chambered heart with two atria and a single ventricle, whereas crocodile, birds and mammals possess four chambered heart with two atria and two ventricles, right? So in fishes the heart pumps out deoxygenated blood which is oxygenated by the gills, but they are not sure. Okay, so in fishes the heart pumps out deoxygenated blood which is oxygenated. उधर से बात करेंगे देखना. Okay, so in fishes the heart pumps out deoxygenated blood which is oxygenated by the gills and supplied to the body parts and supplied to the body part and supplied to the body parts from where deoxygenated blood is returned to the heart. Okay, oxygenated by gills and supplied to the body parts from where deoxygenated blood is returned to the heart. So heart pumps out the deoxygenated blood, okay, which is oxygenated by the gills and supplied to the body parts from where deoxygenated blood is returned to the heart. There is single circulation. So amphibians and reptiles, so they have the single circulations, right? So in amphibians and reptiles the left atrium receives oxygenated blood from the gills, lungs, skin, that is left atrium, and the right atrium gets deoxygenated blood from the other body parts. You will understand this on the diagram, right? However, they get mixed up in the single ventricle which pumps out mixed blood that is incomplete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria respectively passes on to the ventricles of the same side, that is left ventricle and right ventricle. The ventricles pump it out without any mixing up, that is two separate circulatory pathways are present in this organism, and these animals have double circulation because they do not show any type of mixing up. So let us study the human circulatory system. Now the next part is human circulatory system. Then you will see the cardiac cycle, then you see the ECG, then you see the double circulation, then you see the regulation of cardiac activity, then disorders. So this is how it goes, right?